So the aim of this short video is to collect a few of the ideas that we've discussed about interpreting findings from observational studies into one place and provide a loose framework for appraising such studies. So if you read about observational studies and epidemiology in the news, you might be left with the impression that this is how epidemiology operates. So we randomly spin a wheel of fortune for the exposure and the outcome and the target population, and we end up with a story. So in this case, coffee can cause depression in twins. So we have to interpret associations from observational studies carefully, particularly when the question is etiological or we're interested in a causal mechanism. So this is a checklist that's useful when reading about a finding in an observational study, and it's worth remembering this. So could the finding be due to chance, bias, confounding, or reverse causality? So there are alternative explanations for an association that we see. And if we're satisfied that it's not due to that, we then might ask whether the association that we see is causal or not. So let's have a look at each of these in a tiny bit more detail. So first, could it be due to chance? i.e., could it be just random sampling variation that has produced this fluke observation or fluke result? And we try to exclude chance by using hypothesis tests or p-values and by estimating confidence intervals. These two statistics try to give us some information on the uncertainty in our estimate. And there are a list of ways we can avoid being fooled by chance, for example by designing adequately powered studies and by avoiding the sort of binary interpretation of p-values as not significant or significant. We'd also want to see whether a study has done lots and lots of comparisons or subgroup analysis, because if they have, then we'd expect to see some results with small p-values just due to chance alone. And we should always report confidence intervals and look at the size of effect. And if some analysis is exploratory, that should be stated that it's exploratory. And if it is, then we'd probably want to see that those findings have been replicated in another sample. And what about bias? Well, bias occurs due to defects in the design and conduct of a study. It's essentially a distortion away from the true value. And statistics cannot tell us anything about bias, so it's something that we need to remove in the design and to think about when we interpret a study. So we've mentioned quite a lot of different biases in the videos on observational study designs, and you can pretty much group them into two types, so selection bias and measurement bias. Now, more than 50 different types of selection bias have been defined in various papers, but some of the ones that we discussed, some of the bigger ones we discussed here are, for example, in a cross-sectional study, is the sample representative of the target population if we're interested in estimating the prevalence of something? In a case control study, if we're using hospital controls, have we over-represented the exposure in the actual population that generated the cases? It's really the exposure in that group that we're trying to estimate. And loss to follow-up bias, that's another type of selection bias. This is likely to be a problem for cohort studies in particular, how many subjects have been lost over the follow-up period. And measurement bias, we've talked about random measurement error. So that's a bit like white noise that's just been scattered indiscriminately in a measurement or over a variable. The error is not systematically different in the exposure groups or in the outcome groups. But if the exposure is systematically different in the exposure or the outcomes groups, then that's called differential measurement error. And we've talked about two types, recall bias and observer or interviewer bias. So as I said, we can't really do a lot about bias in our statistics or analysis of a study. So it's something that we have to think about in designing and interpreting results. So here are a few questions that we may want to ask ourselves that would enable us to think about bias when we come to interpreting a study. So then there's confounding, which is unwanted sources of variation between our exposure groups. That's also associated with the outcome, leading to a distortion in the association of interest, so either an under or overestimate of the true effect or true association. So if we look at this example here where we're interested in the relationship between vitamin C and birth weight, even if we're able to control for all the potential confounders of this relationship, we'd never be certain that we've accounted for all of the unwanted sources of variation between our exposure groups, because we might have measured some of these confounders imprecisely, or we might have left out some potential confounders or not measured them, and in which case there would still be residual confounding or unaccounted for confounding. And could the association be due to reverse causality? So when the exposure is collected after the outcome has occurred, as happens in a cross-sectional or case control study, then reverse causality might be an explanation for a finding. So lastly, as I've said, if the question is etiological and we've satisfied ourselves with 1, 2, 3 and 4, then we may then ask whether the relationship is causal or not. So hopefully this checklist can serve as a little guide when interpreting associations from observational studies. And to summarise, each type of study has its own strengths and weaknesses that will often depend on the context or the research question being asked. The interpretation of a study then needs to take account of this context and be bespoke to the research question. By understanding how bias, chance, confounding and reverse causality may affect results from an observational study, we can make a better judgment in our interpretation. And to conclude, it's rare that one type of study alone will provide sufficient evidence that a factor is linked to an outcome. Often we want to see that findings are robust and consistent in studies using other designs that are less prone to bias. As such, our interpretation of a study also needs to be put into the context of wider research in the field.